Good morning. Welcome to 8th virtual class, in which we are continuing the chapter called Molecular Basis of Inheritance. We already come across so many topics under this chapter. Now continuing the same chapter, today we are going to learn about mutation and genetic code. If you are remembered, in the previous class we have come across about the genetic code. One of the important concepts, the coded information present in the DNA. Now we are going to learn how this will work. The genetic code and mutation. To understand the relationship between the gene and the DNA, it is the mutation. Mutation is nothing but any alteration in the DNA of an organism which brings about changes in phenotypic and genotypic of an organism. Repeat, it is the alteration in the DNA which brings about the changes in both genotypic and phenotypic characters of an organism is called mutation. So there are two major types of mutation. One is called point mutation. In point mutation, alteration occurs in the form of one single nitrogen base at one particular point or location. That brings about the change in genotype of an organism, which ultimately changes the number of phenotypic characters of an organism. The best example to understand this is sickle cell anemia. In human beings, the RBC, the red blood cell, is having two chains of amino acid, alpha chain and beta chain. In the beta chain of amino acid, the sixth amino acid in the beta chain of amino acids of RBC, the series of amino acids are attached to one another to form a chain. Here the sixth amino acid is happen to be glutamine. But due to point mutation, in the place of this glutamine, amino acid called valine will be replaced. Because of the replacement of glutamine with the help of valine, the RBC, which is having a shape of somewhat biconcave or somewhat disc-like structure, these shapes will turn into sickle shape. All RBCs will turn into somewhat sickle shape. This condition, where RBC turns into sickle shape because of replacement of one amino acid at sixth position. The sixth amino acid is glutamine, which is replaced by valine, which brings about the change in the structure of RBC. Where all RBC turns into a sickle shape, that brings about an anemia called sickle cell anemia. This is called point mutation. Similarly, there is second one called Frameshift mutation. Frameshift mutation. In this, either a nitrogen base, a single nitrogen base, or a pair of nitrogen base will be added, or it will be deleted to the nucleotide sequence. Results, it will shift the frame of the nucleotide sequence, which brings about the changes in an amino acid sequence in the protein. That is what we called frameshift mutation. So either because of addition, it may be shifted forward, or due to deletion of one nitrogen base, it may be backward shift. So both, whether it may be addition or deletion, it will bring about the shifting the position of the nitrogen bases. Brings about changes in an amino acid sequence in the in a chain of amino acids, which is going to be changed in the protein content. So that is what we call the frameshift mutation. We'll take one example. If we consider one statement called, let me describe here. If we considered a statement made up of three words, Ram has red cap. It resembles genetic code. As we know, the genetic codes are made up of triplet nature. So it resembles triplet code here. Ram has red cap. If we add B at the position of red, or between S and R, if we add B here, because of that the frame will be shifted, like we can see here, B, this RE will remain, and this D will be shifting the other side, D, C, A and P. This brings about the shifting of the position. Now the amino acid sequence will be altered. Now if you add one more letter I at this region, that brings Ram has BI, and then R, again it will shift to the forward position, E, D, C, then AP. Then again if we add one more letter in the form of G at this position, see the position, Ram has big R-E-D. This R is shifting here, ED, and the C will be shifting that side, cap. Now earlier, red position was third nitrogen base, that is third triplet code. Because of the addition of one set of nitrogen bases, the triplet here, now the position has shifted to fourth position. Ram has big red cap. Third position was red earlier, now the red has gone to the fourth position. So this is what we called the shift position, or what we call the frame shift mutation. This is one of the common type of mutation which is occurring, in which the frame either it may be deleted or it may be added to the nitrogen bases or in the nucleotide sequence in the DNA, brings about alteration in the DNA, which brings about change in genotype and the phenotype of an organism. That is what we called mutation. Now we will pass to the next topic called RNA. Another one very important concept called tRNA, or also called adopter RNA. tRNA is also called adopter RNA. If you remembered, in the previous classes we have learned there are three forms of RNA: mRNA, tRNA and rRNA. All these three forms of an RNA, that is mRNA, tRNA and rRNA, is going to be synthesized from DNA by means of a process called transcription that we already learned in the previous classes. Now, one of the form of an RNA called tRNA we are going to learn today. RNA is all the forms, whether it may be mRNA or tRNA or RNA, it is made up of single polynucleotide chain that we all learned in the very first class. DNA is made up of 2 polynucleotide chain. RNA is made up of single polynucleotide chain. As it is made up of single polynucleotide chain, no base pairing. Single strand, no base pairing is found in an RNA. Now we are going to learn about tRNA. tRNA is a form of RNA which synthesized from DNA by means of transcription, which is also called adopter RNA. The adopter RNA is because this will have very important role to read the codons present on mRNA. Another one, it will going to attach to amino acids. It is having dual role, dual function. One side it will read the codons present on mRNA, and other side it will bind to the amino acid, which is for the purpose of polymerization of amino acids to form protein. Now, what is that tRNA? So, as tRNA is single stranded, but it is highly folded. As it will be folded, it is bringing the strands or the ends of the tRNA closer to one another. It is the Francis Crick. Might have been remembered, Francis Crick and James Watson, the proposed DNA double helical model. So the same scientist by name Francis Crick postulated the structure of or explained the structure of tRNA, where the tRNA resembles clover leaf. There is one leaf called clover leaf. So here the tRNA resembles clover leaf, where RNA is highly folded. As a result, the two ends are coming closer to each other. So the Francis Crick has explained the clover leaf model of tRNA, in which tRNA show 5 arms, in which the 2 strands are coming closer to each other. The 2 ends of the tRNA coming closer. This is 5' end, this is 3' end. There is one end called anticodon site, and the 3' end, it is ending with CCA. This is called amino acid binding site. I'll repeat. The tRNA which resembles clover leaf because of its highly folded condition, the 2 ends are coming closer to each other. Because of that, it will appear in the form of a somewhat clover leaf. and it shows two important ends. As I mentioned in the beginning, it reads the codons. As we know, the codons are the genetic form of information present on mRNA that will be read by tRNA with the help of anticodon or NODOC region. This area of tRNA or this site of tRNA reads the codes present on mRNA. Like suppose AUG is present on mRNA. The code present in tRNA is in the form of an anticodon which is complementary to the codon present on mRNA, that is UAC. This complementarity or the base pairing is one of the major features to read the codes present on mRNA. Similarly, the other end, opposite end of this is called amino acid binding site. As we know, there are 20 different types of amino acids are present. To carry each amino acid, there is a particular tRNA will going to bind to the particular amino acid. For that, the particular code will be present in the form of an anticodon that will going to read the codes present on mRNA. So there are two functions here, dual function. As I told, it is having dual function. One, it reads the codon, other one, it is attaching to the amino acid. Apart from that, it is having the two arms. This is called enzyme. It is called enzyme binding site. It is called enzyme binding site. One end is called enzyme binding site. The opposite end is called ribosome binding site or ribosomal attachment site. Ribosome site, enzyme binding site, which will help out for the process of translation. There is nothing but the protein synthesis. I'll repeat to conclude this. The tRNA is one of the form of RNA which is highly folded, brings the two ends closer to each other because of its folded nature. It resembles clover leaf, bearing the 5 important arms. One is called the amino acid binding site, where amino acid will going to attach to the tRNA, and the corresponding codes will be found on the opposite end called anticodon site, which will going to read the codes present on mRNA. Whereas the other two arms found on other side, one is the enzyme binding site or enzyme recognizing site, and other opposite end is called as a ribosome binding site, which play a role for the protein synthesis. Apart from that, there is one extra arm which is not having any specific role. This RNA, which resembles clover leaf, and that was proposed by Francis Crick in the form of a model called clover leaf model, which is going to work for the process of protein synthesis. Now, the protein synthesis is another very, very important event. Is going to be carried out with the help of mRNA, tRNA, and rRNA. So the whole process of protein synthesis, let me describe protein synthesis is one of the most important metabolic event which is guided by DNA, which plays a role to synthesize protein in every cell. It is totally guided by DNA. DNA, as we already discussed in the previous classes, DNA involves in a synthesis of RNA. That event is called transcription. This RNA, which is of three different types, mRNA, tRNA and rRNA, that involves in a process called translation. It is the process of protein synthesis which is totally guided by DNA. It is called DNA dependent event. So in the first stage, DNA will going to involve in synthesis of three forms of an RNA called mRNA, tRNA and rRNA by utilizing three forms of DNA polymerase enzymes. Remember, DNA polymerase 1, polymerase 2, polymerase 3, which show the division of labor in all the eukaryotes, resulting it will form three different forms of an RNA, and this whole event of synthesis of RNA from DNA is called transcription. And this transcription is totally regulated by RNA polymerase enzymes, which are of three different types: polymerase 1, polymerase 2, polymerase 3, which is dependent on DNA. They are commonly called DNA dependent RNA polymerase enzyme, which transcribes the three forms of an RNA. So as we have three forms of RNA polymerase enzyme, RNA polymerase 1 synthesizes one form of RNA, RNA polymerase 2 synthesizes one more form of RNA. Like that, all the three forms of RNA polymerase enzyme involves in a transcription of three forms of RNA. Now these three forms of RNA along with the amino acids involves in a process of translation, which ultimately gives the product called protein. So all these three with amino acids with several enzymes will work together for a process of protein synthesis, that is by means of an event called translation, that we'll learn in the next class.